CRISPR-Cas9 is a revolutionary gene editing technology that has transformed the field of molecular biology and genetic engineering. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it was first discovered in bacteria, where it serves as a part of their immune defense against invading viruses. Scientists realized they could repurpose this system to precisely edit DNA in other organisms, including humans. CRISPR uses a specially designed RNA molecule to guide the Cas9 enzyme to a specific location in the DNA sequence. It finds the target. Cas9 acts like a pair of molecular scissors and cuts the DNA at that precise spot. The cell then tries to repair the break, and that's where scientists can introduce changes, such as fixing a genetic mutation or adding a new gene. This tool is now being explored to treat genetic disorders like sickle cell anemia, muscular dystrophy, and even some forms of blindness. It's also used in agriculture to create stronger, more resilient crops. However, CRISPR also raises big ethical questions. Should we edit human embryos? Could this lead to designer babies? As powerful as it is, CRISPR is still a tool under careful study, and it might just redefine the future of medicine, food, and evolution itself.